This presentation is on Listeria monocytogenes and how the immune system responds to this infection. Listeria is a gram-positive, non-spore-forming bacillus. This pathogen is associated with soil, sillage, water, foods, and food processing environments. It is highly tolerant to many different conditions, including being psychotolerant, acid-tolerant, and salt-tolerant. The serious infection that is caused by this foodborne pathogen is known as listeriosis. Some of the common foods that can become easily contaminated with listeria are queso fresco and other soft cheeses, raw sprouts, melons, hot dogs and deli meats, smoked fish, and anything involving raw milk. This pathogen can be found in soil and water and thrives in a moist environment. Vegetables can become contaminated from the soil or manure used as fertilizer. Animals can also carry listeria and can contaminate food products. From there, the food is processed and consumed by humans. Because contamination can come from anywhere, it is difficult to determine the source of these outbreaks. Contamination is very dangerous and should be limited as much as possible. While there are only about 1,600 cases in the US each year, about 90% of individuals are hospitalized and the death rate is between 20 to 30%. The infectious dose of this pathogen can also be less than 1,000 cells per susceptible individual. While listeria does not typically lead to infection of healthy individuals, those who fit within the risk demographics, such as pregnant women, those over the age of 65, immunocompromised individuals, and infants, have a high chance of developing listeriosis, which can also lead to death. Next is the immune system's response to listeria. The innate response to listeria begins with neutrophils. They are important in the initial control of listeria growth due to reactive oxygen and nitrogen intermediates, which ultimately kill extracellular bacteria. Some studies have even shown that as neutrophils generate these intermediates, this also increases the mediated killing that macrophages perform. These neutrophils secrete cytokines such as IL-6 to secrete chemokines, including CSF1 and MCP1 at the local site of infection. These chemokines signal macrophages to this site in order to continue fighting the infection. But macrophages are the main source of innate immunity during listeriosis because this pathogen first localizes within the macrophages in the marginal zone. When concerning listeriosis, macrophages allow the pathogen to escape from the phagosome in order to activate certain signals like the neutrophils and their intermediates. These macrophages are then able to secrete cytokines TNF-alpha and IL-12. Both of these drive the secretion of NF-kappa B transcription factors, which then signal to produce IFN-gamma. The secretion of this interferon neuron leads to activation of more macrophages to increase their bactericidal activity. Other interferon genes are also involved within the macrophage killing of listeria, such as p47 GTPases, but the mechanisms and how this is done is still undetermined.
B cells and antibodies don't play a major role in the immune response against Listeria monocytogenes, and this is mainly because the majority of bacteria in this infection are found inside the cell, and antibodies act on things that are extracellular. Antibodies in this case can function to neutralize a pore-forming virulence factor of Listeria that's called Listeria lysine O, or LLO for short, and this allows the bacteria to escape the phagosome, and IgG would be the most effective subtype for neutralizing LLO. B cells have also shown to be important in the maintenance of memory CD8 T cells that are generated during the initial immune response with Listeria.
CD4 and CD8 T cells comprise most of the adaptive immune response to Listeria. Proteins from Listeria residing in the host cell's cytosol are degraded by the host cell's proteasome. The peptide fragments are transported to the endoplasmic reticulum by the protein tap, loaded onto MHC class I molecules, and presented on the cell surface to CD8 T cells. The CD8 cytotoxic T cells directly kill the cells infected with Listeria by secreting perforin, which forms small pores in the cell's membrane to help granzyme get into the cell. Granzyme then activates the infected cell to apoptose. Professional antigen-presenting cells also present antigen from Listeria destroyed in lysosomes via the MHC class II pathway to CD4 T cells. Antigens from the lysosome can also traffic to the endoplasmic reticulum, where they are presented by MHC class I molecules through cross-presentation by dendritic cells. CD4 helper T cells do not directly kill the infected cells, but instead secrete cytokines to activate macrophages to improve their immune function. Listeria induces a strong Th1 CD4 T cell subtype response. Th1 cells secrete interferon gamma, which aids macrophage activation. Interferon gamma makes macrophages better at killing Listeria by increasing their killing mechanisms, such as reactive oxygen species and nitric oxide. Macrophages phagocytize Listeria and also increase their expression of MHC molecules and increase the co-stimulatory molecule B7, which helps to activate more T cells to support the immune response.
Memory T cells have a lower stimulation threshold for activation and upon secondary infection can quickly exert effector functions, allowing rapid control of infection and elimination of the bacteria. Memory T cells swiftly mobilize to the site of infection and secrete IFN gamma, leading to activation of macrophages, and then rapidly secreting molecules which are directly toxic to infected cells such as perforin and granzyme. Memory T cells are essential for protection from reinfection with Listeria due to their ability to quickly recognize and destroy infected cells.
Another type of T cell is the regulatory T cell. It isn't well understood what factors cause the development of regulatory T cells yet, but we do know that their development can occur both during negative selection in the thymus or during activation in a secondary lymphoid follicle. These T cells are responsible for limiting the immune response by secreting immunosuppressive cytokines like TGF-beta and interleukin-10. And from what we currently understand, regulatory T cells don't play a major role in the initial infection with Listeria, but they can play a role in limiting the expansion of memory CD8 T cells if there's reinfection.
Now we will discuss how Listeria evades the immune system.
A bacterial pathogen capable of hiding in a host cell initially enters by endocytosis. Here, the pathogen induces the host cell to take it up by phagocytosis and ends up in a phagosome. These intracellular pathogens, which live inside host cells as unwanted guests, gain temporary safe harbor within the phagosome. This is because the antibodies within phagocytic cells will not penetrate the host cell. Once inside the phagosome, intracellular pathogens have three options to avoid being killed by the fusion of the phagosome with the lysosome, which is full of digestive enzymes. A single pathogenic species can only use one of these three mechanisms. First, some pathogens can simply escape from the phagosome before fusion occurs. Pathogens such as Shigella and Listeria use this strategy and then undergo unrestricted growth in the cytoplasm of the host cell. These pathogens form actin tails, which allows them to move within the cell and even to move into adjacent cells where they continue the infection. These microbes can spread from cell to cell without ever encountering the extracellular environment where they would be vulnerable to attack by the immune system. Alternatively,
Despite the various immune responses to Listeria, Listeria has evolved mechanisms to evade the immune system. Listeria evades the immune system by escaping the phagosome. Upon phagocytosis by macrophages into the body cells, Listeria is capable of avoiding destruction in the vacuole by secreting a pore-forming protein called LLO. LLO forms pores in the phagosomal membrane and lyses it, and thus is able to escape destruction in the phagosome and can enter into the cell cytosol. Shortly after entry into the cytosol, Listeria induces the polymerization of host actin filaments and uses the force generated by actin polymerization to move and migrate to other cells. ACT-A, a bacterial protein, is responsible for mediating actin nucleation and actin-based motility that largely contributes to Listeria's pathogenicity. The study of the immune response to Listeria has demonstrated how incredibly intricate and complex the immune system is and how it can mount a fine-tuned response to virtually any pathogen it encounters.